Now the other big man in this category is sphingolipids. Now remember we say that in sphingolipids, sphingosine is the alcohol which is present, not glycerol. So here we have glycerol, OH group, OH group, OH group, but this is found in glycerophospholipids, not in sphingolipids. In sphingolipids, the molecule defined is sphingosine. It is similar to glycerol because if you look at carbon 3, it has an OH group and 2 hydrogen. Carbon 2, instead of an OH though, it has an NH2. Carbon 1, instead of just an OH, it has a long chain. This chain here with this double bond over there. And this is known as a sphingosine. So sphingosine, the backbone, sphingolipids, the backbone is sphingosine and not glycerol. Okay. Now the major sphingosines we would deal with would be sphingomyelin, cerebosides, and gangliosides. When a fatty acid is attached to the second carbon, a ceramide results. When a fatty acid attaches to this NH2 group, we have a ceramide. And in fact, sphingomyelin, cerebosides, and gangliosides are all types of ceramides. Because you don't just find them with whatever attached here. First, you have to have that fatty acid here, the ceramide, and then these substances can form. And here we have a diagram of a ceramide. But note well that this is the carbon 1 chain, this is carbon 2 where the fatty acid chain is attached, this is carbon 2, a ceramide. A ceramide containing a fatty acid with 30 carbons long is a major component of the skin and regulates skin's water permeability. Remember that. And ceramides, as we were just speaking about, are the backbones of sphingomyelin, cerebosides, and gangliosides. We'll go into a little more details with those. Sphingomyelin, we have a molecule here. If you notice, we have a hydrocarbon chain. Sorry, we have a sphingosine backbone in red. Remember in the second carbon we said a fatty acid is attached, making it a ceramide to the NH2 of the backbone. But on the third carbon we have phosphate and choline attached. When we have a phosphate and choline attached, we have sphingomyelin. So you see this phospholipid is similar to phosphatidylcholine. The difference is that the backbone is sphingosine and not glycerol. The polar head group comprises phosphocholine or phosphoethanolamine, and as I was saying, it's different from phosphatidylcholine. Sphingomyelin, as the name suggests, is a major part of plasma membrane and the myelin sheath. It is found in red blood cells and is a good source of phosphoric acid in body tissue. We were speaking about phospholipids C and D that would cleave here and here. So I would assume that the cleavage would result in the release of phosphoric acid. That is such as an assumption. Cerebosides. Cerebosides want to have a backbone of sphingosine, fatty acid attached to carbon 2, making it a ceramide. But on carbon 3, we have a carbohydrate, in fact a monosaccharide. We have a diagram of glucose. Cereboside where the monosaccharide is glucose. Now they are neutral and they comprise galactocerebosides, which is prevalent in neuronal cell membranes, and glucose cerebosides, which would be found in membranes of peripheral tissues. Now, a sulfolipid is found in white matter, brain, and testicle, and that's where we have, as well as in the chloroplast, and that's where sulfur, if this was a galactose, we would find a sulfur attached. That would make it a sulfolipid. Now globosides, similar to cerebosides, only thing that they contain 2 or more sugars. And like cerebosides, they have no charged pH cell. Now the third class of ceramides, gangliosides. And if you notice, remember a lot of these things names we give them as gangliosides are prevalent in ganglia, grey matter in the CNS, as well as in the spleen. They are derived from ceramide oligosaccharides. Those are carbohydrates slightly longer chain, and they contain a sialic acid residue, and this sialic acid residue will make them slightly acidic. Gangliosides, very important, they function in cell recognition, and they give rise to ABO blood groups. So this is very important. So next time you hear about ABO blood groups, remember gangliosides. Now this is just a table giving a brief summary of what we've been talking about. Here we have the sphingosine backbone. If just a fatty acid is attached to the second carbon where the nitrogen is present, we would call it a ceramide, and here the X would be a OH. If phosphocollagen is attached, we call it a sphingomyelin. If a glucocereboside or a galactocerebide is attached, we call it a cereboside. If a glucose or a galactose is attached, we call it a cereboside. And if you have di-tri-mono, we call it a globoside. And if you have oligosaccharides attached, we call it a ganglioside, GM2 in this case. And here we see any 5-8 that's representative of N-acetylminic acid, which is a type of sialic acid, as we mentioned previously. Now glycerophospholipids and sphingolipids are degraded in lysosomes, so it helps to maintain the balance. They're being produced and they're being degraded to maintain the balance. And the hydrolytic enzymes involved are phospholipids. So once again, I reproduce this diagram showing the action of phospholipids degrading these phospholipids. And it's very important that you notice that hydrolytic enzymes, anything where you see hydrolysis, hydro water means the splitting in the presence of water. So in order to form the ester bond, remember we lost a water molecule to form this ester. So to split it, we add this water molecule and hydrolyze the bond. This is what these phospholipases do. Okay, and that's how these phospholipids are degraded. Wow! So you've done fatty acids, you've touched on epoisinoids, triglycerides, waxes, glycerophospholipids, and sphingolipids. So now the final category that we'll be dealing with will be the isoprenols. Now this is an isoprene molecule consisting of 5 carbons, 1, 2, 3, 4, 5. Now isoprenols may be divided into terpenes. This is a large group of compounds made of repeating isoprene units. They're found in essential oils, resin oils, and rubber. And it's important that we realize that vitamin A, E, and K are either included or closely related to terpenes because of this isoprene. So that's terpenes. Steroids. Steroids. This is the general structure of steroids, consisting of 4 ring structures. 4 of them are 6 carbon rings, and 1 of them is a 5 carbon ring. We usually call it ABCD, and the OH group is attached to this one. Now this is a diagram showing cholesterol. See the general structure here. Most important to realize it on the 10th and 13th division. You will find methyl groups attached, but in cholesterol on the 17th portion, you will find this chain attached. Now steroids are synthesized from 5 isoprenol units in eukaryotes. And sorry about that. And due to the hexagonal rings, many isomers may form. Now, if you did a little background chemistry, you would know that when you have a hexagonal ring or rings of these structures, they may form what are known as chair or boat isomers. These are different configurations which should have varying degrees of stability. But the mere fact that you have 5 different ring structures here means that a lot of variation occur. And it's important to realize that these rings are not benzene rings. They are not benzene rings. You know, benzene rings you see the circle indicating alternating double bonds in the resonance structure. But these are not benzene rings. And the bullet in note where the metal groups are attached and the cholesterol at this site. Now sterols are the precursors of steroids, bile acids, adrenocortical hormones, sex hormones, and vitamins, and cardiac glycosides. And if you see one bile acid here, you see it's similar to that of cholesterol. And in fact, a lot of times cholesterol is broken down to form bile acids. So no one of the structures are similar. Okay? And steroids lock the alkyl chain attached to position 17. So in order to be a steroid, you have to move its alkyl chain. These are sterols, but in order to be a steroid, you have to remove the alkyl chain from position 17. And as a result of moving this long hydrocarbon chain, you would find that steroids are more polar than cholesterol. Note 12. Prednisone and prednisolone are steroid drugs with potent anti-inflammatory activities. We've done with terpenes, we've done with steroids. Time to move on to lipid vitamins. Now lipid vitamins are made from repeating isoprenol units once more. They are the fat-soluble vitamins ADEK, A, D, E, and K. Now here we see the general functions of each of these vitamins. A, they form the visual pigment in the eye of vertebrates, and they regulate gene expression during epithelial cell growth. And vitamin D is a precursor to a hormone which regulates calcium metabolism. That's right, it's very important for babies. So go for their early morning walks, because without the hormone, how could you have calcium metabolism? And vitamin D here we see is a precursor, so it must be there before this can go on. So when you see a baby with bowed legs, it is because the bone is not formed properly. It's the result of a lack in vitamin D, which is the precursor. So that. Now vitamin E is an antioxidant, you know. A lot of times in cells you will find free radicals, free oxygen radicals attacking the membrane. But vitamin E is a very important antioxidant. And vitamin K, as we may have seen before, is essential in the blood clotting process. Alright, so these are the general lipid vitamins. Wow! So we've done fatty acids, we've done glycosides, we've looked at triglycerides, we've touched on waxes, we've looked at glycerophospholipids and sphingolipids. We've touched on isoprenols, terpenes, steroids, lipid vitamins. Wow! We've done with the general description of lipids. We've gone through the classification of lipids. We looked at the structure and the function of lipids. I really hope that by the end of this presentation you have a thorough understanding about the basics that you should know concerning lipids. Christopher Hill. Adios. Fat. It's finished. Wow.